Hello, welcome to ECDC, Majlis Arts and Science College, Puramannur. Myself Janina Mariam from PG Department of Microbiology, and I am here with my topic glycolysis. There are several hundreds of reactions taking place in living cells in a well-organized and integrated manner. The entire spectrum of chemical reaction taking place in cells are collectively known as metabolism. The term metabolic pathway constitutes a series of enzymatic reactions to produce specific products, and the term metabolite is applied to a substrate or an intermediate or a product in the metabolic reaction. Metabolism is broadly classified into two categories, that are catabolism and anabolism. Catabolism is a degenerative process concerned with the breakdown of complex molecules into simpler ones, while anabolism is a biosynthetic process involving formation of complex molecules from simpler ones. Here comes the main metabolic pathway, glycolysis. The term glycolysis is derived from two Greek words, that are glycos and lysis. Glycos means sugar or sweet, while lysis means dissolution. It is considered as a universal pathway in all the living cells. It is also known as EMP pathway, referred to as Embden-Meyerhof-Parnas pathway, which are the biochemists behind the discovery of glycolysis. And glycolysis is defined as the sequence of reactions converting glucose into pyruvate or lactate with the production of ATP. Salient features of glycolysis. There are 7 salient features in glycolysis. First one is glucose or glycolysis takes place in all the cells of the body because the enzyme of this reaction is present in the cytosomal fraction of the cell, and every cell has cytosome. Second one, glycolysis takes place in two ways, in aerobic condition and anaerobic condition. In anaerobic condition, lactate is the end product, while in aerobic condition, pyruvate is the end product, which is then oxidized to carbon dioxide and water. And third one is glycolysis is a major pathway for ATP synthesis in cells that are lacking mitochondria, example erythrocytes, cornea, lens, etc. Fourth one, glycolysis is very essential for brain, which is dependent on glucose for energy. Here glucose in brain is first undergoing glycolysis before it is oxidized to carbon dioxide and water. And the net reaction for anaerobic glycolysis is summarized as glucose plus 2 ADP plus 2 Pi giving 2 lactate plus 2 ATP. Then the sixth important salient feature is glycolysis is a central metabolic pathway with many of its intermediates providing branch points to other metabolic pathways. So, intermediates of glycolysis are useful for the synthesis of amino acids and fats. And the seventh point is reversal of glycolysis along with alternate arrangements of its irreversible pathway leads to the production of glucose, and that process is known as gluconeogenesis. Coming to the reactions of glycolysis, there are 10 reactions in glycolytic pathway. Among the glycolytic pathway can be divided into 3 distinct phases. First one is energy investment phase, second one splitting phase, and third one energy generation phase. This is the glycolytic pathway mentioning the enzymes and products on the right side of this picture. Coming to the stepwise details of glycolysis, step 1 is phosphorylation, where glucose is phosphorylated by ATP to form glucose 6-phosphate, and this is an irreversible reaction catalyzed by the enzyme hexokinase, and the reaction can be represented as glucose converted to glucose 6-phosphate by the phosphorylation using ATP and with the help of enzyme hexokinase. Step 2, isomerization. It is a reversible reaction with arrangement of chemical structures of carbonyl oxygen from C1 to C2 forming a ketose from the aldose. Here the aldose glucose 6-phosphate gives the ketose fructose 6-phosphate, and the equation can be written as glucose 6-phosphate converted to fructose 6-phosphate using the enzyme phosphoglucoisomerase. Phosphorylation. Here fructose 6-phosphate is phosphorylated by ATP to form fructose 1,6-bisphosphate. This is an irreversible reaction which is catalyzed by an important regulatory enzyme phosphofructokinase, and the reaction can be summarized as fructose 6-phosphate getting converted to or phosphorylated by the ATP to fructose 1,6-bisphosphate with the help of the enzyme phosphofructokinase. Step 4. Splitting phase. Here 6-carbon compound is cleaved to form two 3-carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and this reaction is catalyzed by the enzyme aldolase. Here fructose 1,6-bisphosphate is converted to two molecules of 3-carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Step 5. Isomerization. Dihydroxyacetone phosphate is isomerized or oxidized to form glyceraldehyde 3-phosphate using the enzyme triose phosphate isomerase, and here is the change in glyceraldehyde 3-phosphate isomerized to dihydroxyacetone phosphate, or dihydroxyacetone phosphate is changed into glyceraldehyde 3-phosphate. Here we get two glyceraldehyde 3-phosphate molecules. Step 6. Two molecules of glyceraldehyde 3-phosphate is oxidized, and glyceraldehyde 3-phosphate dehydrogenase is the enzyme which oxidizes or catalyzes the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Step 7 is phosphorylation. The transfer of high energy phosphate group that was generated from ADP to form ATP. This phosphorylation, that is addition of phosphate group to ADP to give ATP, is known as substrate level phosphorylation, and as the phosphate donor in this is the substrate 1,3-bisphosphoglycerate 1,3-BPG, and the product in the reaction is 3-phosphoglycerate, and it can be summarized as 1,3-bisphosphoglycerate getting converted or phosphorylated to 3-phosphoglycerate by the addition of phosphate group from 1,3-bisphosphoglycerate to ADP to form 2-ATP, and the enzyme involved in this process is phosphoglycerate kinase. This is the first substrate level phosphorylation taking place in glycolysis. Step 8 include the remaining phosphate ester linkage in 3-phosphoglycerate is moved from 3rd carbon to 2nd carbon because of its relative low free energy of hydrolysis to form 2-phosphoglycerate, and it is summarized as 3-phosphoglycerate converting to 2-phosphoglycerate using the enzyme phosphoglycerate mutase. The step 9 involves dehydration of 2-phosphoglycerate. This is the second reaction in glycolysis where a high energy phosphate compound is formed, and that compound is phosphoenol pyruvate or PEP. The 2-phosphoglycerate is dehydrated by the action of the enzyme enolase to form phosphoenol pyruvate or PEP, and this is a reversible reaction. It is summarized as 2-phosphoglycerate converting to high energy phosphate compound phosphoenol pyruvate with the help of enzyme enolase where there is a removal of water molecule. The last step is transfer of phosphate from PEP to ADP. That is, this last step is a reversal transfer of high energy phosphoryl group from PEP, that is phosphoenol pyruvate, to ADP molecule, and this reaction is catalyzed by a powerful enzyme pyruvate kinase. This is the second substrate level phosphorylation which is taking place in glycolysis which yields ATP, and this is a non-oxidative phosphorylation reaction, and it is summarized as phosphoenol pyruvate getting converted to pyruvate using the enzyme pyruvate kinase, and here occurs the second substrate level phosphorylation with formation of ATP. Here comes the end to the reactions of glycolytic pathway. Now let us discuss about overall balance sheet of glycolysis. Each molecule of glucose gives 2 molecules of glyceraldehyde 3-phosphate, and therefore total input of 10 reactions of glycolysis can be summarized as glucose plus 2 ATP plus 2 PI plus 2 NAD plus 2 H plus 4 ADP giving 2 pyruvate plus 2 H plus plus 4 ATP plus 2 molecules of water plus 2 NADH and plus 2 ADP. And on cancelling the common terms from the above reaction, we can get the net equation for glycolysis, that is glucose plus 2 ADP plus 2 PI plus NAD plus giving 2 pyruvate plus 2 NADH plus 2 ATP plus 2H plus 2 molecules of water. Energy yield in glycolysis. It occurs in first step where glucose is converted to glucose 6-phosphate. There is a consumption of 1 molecule of ATP. In second step, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. There is also consumption of 1 molecule of ATP. And in the seventh step, 1,3-bisphosphoglycerate is converting to 3-phosphoglycerate. There is a gain of 2 molecules of ATP. And during the last step where PEP is converted to pyruvate, there is a gain of ATP of 2 molecules. So the net gain of ATP is 4 minus 2, that is 2 molecules of ATP. Regulation of glycolysis. Among the 10 reactions of glycolysis, 3 reactions are irreversible, which are catalyzed by the enzyme hexokinase, phosphofructokinase and pyruvate kinase. They drive the glycolysis cycle and are important in the regulation of glycolysis. First one, hexokinase. They are involved in the phosphorylation of glucose, which is inhibited by its product glucose 6-phosphate, because in order to slow down the glycolysis, glucose 6-phosphate inhibit hexokinase activity. And the second one is phosphofructokinase. It is a major regulatory enzyme and considered as the rate limiting committed step in conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. It is an allosteric enzyme with allosteric effectors and allosteric regulators. The activators for this enzyme is ADP, AMP and fructose 2,6-bisphosphatase. Then inhibitors are ATP and citrate ions. The third one is pyruvate kinase. It is an allosteric tetramer enzyme which is activated or active in its dephosphorylated state and inactive in its phosphorylated stage. The inhibitors for this enzymes are ATP, acetyl-CoA and fatty acids, while the activators for this pyruvate kinase enzyme is fructose 1,6-bisphosphatase. And that's all for glycolysis. Please check out the description below and thank you for watching.